Hey guys, here we are today, Monday, May 4th, and we are talking about the Nephron. I think you're supposed to say on May 4th, and may the 4th be with you. I know if we were at Great Falls High today, Mrs. Mater would be wearing her Darth Mater outfit in the hallway, so I'm missing those fun shenanigans that we get into. Cinco de Mayo's tomorrow. All right, well, we are talking very quickly today about how blood is filtered in the kidney, a little bit about filtration, secretion, and reabsorption. We will get more into that later this week. And I am not in the office today. I'm here on my sunny porch enjoying a little bit of vitamin D coming in through the windows here. So change of scenery a little bit. All right, so we are on page 3 of your study guide packet, and I would recommend having colored some things, pens or colored pencils or highlighters work great too, to color coordinate some of these little drawings we're going to do. First thing we're going to do before we label this guy is right here it says sketch a simple nephron below. And so you can see it's a very, very simple drawing. On the screen here, the one I made is colored instead of just in pencil. Any way you want to do it is great. So that's going to go right there underneath the picture, but we're going to go through it in a little bit more detail here. So as you draw this out, okay, I'm using red to represent my blood vessels, and yellow here is representing those things that are the collecting tubules that are collecting what will eventually form urine. Some of the stuff that ends up in here is good. Some of the stuff that ends up here is bad. We want to eventually make sure it's all bad stuff leaving in our urine eventually. So when you're drawing this out, as you're drawing these blood vessels, I did intentionally make this one a little bit thicker and this one a little bit thinner. The one coming in here, we've mentioned already before, is the afferent arteriole. And for our quick sketch, just use letters to abbreviate these things. We'll label them on the next picture, fully spelling them out. So afferent arteriole is coming in. Now, this little mess, kind of where it all sort of tangles up in the inside, that is the blood vessels, is called the glomulus. And it's just like a big glob of blood vessels, and the glomulus is all globbed up in there. And then leaving that is the efferent arteriole, So just like in the spinal cord, afferent was going towards the spinal cord, efferent away. Coming into the glomulus is the afferent, and leaving is the efferent. E for exit. That will help you. Catching all of the stuff that gets filtered out of the glomulus right here, I fondly refer to it as the catcher's mitt, the Bowman's capsule, BC. And then this portion of the collecting tubules in the simplified drawing that we have here, in a complex drawing, it looks like a hot mess of a bunch of curly, weird tubes. So to simplify it out, we've just shown it as some little squiggly lines, all sort of like if I could stretch out this thing, this is what it would look like. So we've got here our proximal collecting tubule because it's proximal or closer to my Bowman's capsule. We also have in here the loop of Henle, LH, and then the distal collecting tube further on. Distal because it's further from the Bowman's capsule. But what it is close to is the collecting duct, and that's going to eventually end up with all the urine and excreted substances coming out of it. Why we draw this simplified picture instead of having you sketch this is just like when we did the heart. If you remember, we drew a box and divided it into 4 chambers to represent the chambers of the heart. To understand the physiology, sometimes it's easier to look at a simplified version of it instead of looking at this picture, just to understand sort of where things are in the sequence of events. So this will hopefully make sense further on. Okay, so as we go through this picture here, if you have the black and white version, I would recommend maybe coloring and color coordinating your page along with what you see on the screen. I just took like a pen and like briefly outlined these things with my pen, just so I could see where these different colors are, and it catches my eye a little bit better. If you print it off the colored version, then you're already good to go. So we're going to talk about 3 major processes on here, and then they are identified on here with the colored arrows. The colors of these aren't important, but I would keep with the red and the purple and kind of a yellow for the other structures. So 3 processes that we see occurring in the nephron. First process is filtration. That happens at the Bowman's capsule. And then the next process is reabsorption. And then the third process is secretion. All 3 of these things are important for getting the good stuff back into my bloodstream and the bad stuff out. That's the goal. We want to urinate out the bad. We want to keep the good. That is the goal of all of this. So let's go through some of these things, most of which we've already labeled on a previous picture, but now this is in a different view, in a different form of that. So this is that blood vessel that brings the blood to my nephron, that cortical radiate artery. This is the one that was heading up in the renal cortex of my kidney when we were talking about the outside of the kidney. That's where these blood vessels are. The little small branches that branch off are arterioles. And notice, this is a little thicker coming in and a little thinner going out. We have here, right, our afferent arteriole going in to our glomerulus, where we've got sort of these different branches of the arteriole, and then leaving on the thinner diametered arteriole is the efferent arteriole. Now, what happens here, because we're going from a wider tube to a smaller tube, is pressure gets high. And when pressure gets high in here, a natural passive process is going to occur, which is, as you see down here, filtration. And so color that AN, whatever you have down here, so that it stands out to you. So filtration is occurring here in the Bowman's capsule. That's our little catcher's mitt that we have there, the Bowman's capsule. So as we have the filtrate coming out, what ends up leaving our blood vessel here is basically what we find in our blood plasma. So if you can think back to our blood unit, blood plasma does not include the red blood cells and white blood cells, but it does include everything else that's floating around in that mostly water liquid. So electrolytes and glucose, and then there's proteins and nucleic acids and some fats, and all of these things just free floating around, hormones. When we go through that first step of filtration in the Bowman's capsule, the good and the bad stuff get filtered out. And so I want you to write good and bad right there. And what that good and bad is filtering into, it gets collected into this tube. Now, this tube twists and turns, and that was what, like, all of this craziness is over here. But on the simplified picture, this tube is representing all 3 parts of the nephron: the proximal collecting tubule, the loop of Henle, and the distal collecting tubule. Okay, so this is just representing that whole twisty, turny mess. Because all of this good stuff has just left my bloodstream, I should say all of the good stuff except for red blood cells, white blood cells, and proteins. Proteins are too big to get forced out of this high pressure system. I need to make sure here at step B, the good stuff gets reabsorbed. I want to reabsorb good. And what it reabsorbs into here is the peritubular capillaries. And those are the capillaries that twist and turn all around my collecting ducts. So what is here, what is in here is the filtrate. I filtrate the good parts of it, get reabsorbed into my peritubular capillaries, and the bad stuff that maybe didn't get filtered out here and made it all the way down here. Here's some bad stuff left over. Things like toxins or alcohol, they get secreted out. They have one last chance to get secreted out here. And so that's step 3. So at the very end, at the very, very end here at step 8, hopefully what I have leaving my nephron, going back into circulation, into the veins, is clean blood. And so all of the bad's been removed from it, just the good remains. And I got clean blood going back into circulation. And urine should be full of all that bad stuff and hopefully none of the good stuff. All right, that is our lesson for today. Go ahead and do your short homework assignment. Later this week, we will get more into the details of what is getting filtered, reabsorbed, and secreted out and where in our nephron. Enjoy your May 4th, and may the 4th be with you. See ya.